Thank you. I'm Ravi, Jonnal. I'm from UC Davis. My lab is in Sacramento, which is why I am said to have hailed from Sacramento. The medical center at UC Davis is in Sacramento, and my lab is in Sacramento. I work on humans. The title of my talk looks like a misspelling of electroretinography, but it's not. I'm going to talk to you about a technique that I've been working on for 15 years, really, that I view as a kind of complement to existing functional tests or tests of retinal function, namely using light and imaging in order to measure neural responses in the retina. Before I start with any of the science, I want to just acknowledge the people who, everyone says like, oh, these are the people who did the project, but I really mean it. These are the people who did the projects. So I work very closely with Robert Zawadzki. He does in animal models a lot of what I do in humans. We were both mentored by Jack Werner, whom I'm sure many of you know, and he's emeritus at UC Davis now, but he's still around. We take our hardest questions to him. We have a couple of project scientists and some postdocs in the lab who've been in the lab for only a few months and are about to make contributions to the projects. But mainly what I'm going to be telling you about is work done by Mehdi Azimipour, Denise Valente, and Kari Vianola, these three down here. So this bottom row is alums. I'd also like to acknowledge our sources of support. Like Omar, I have too many slides and a plan to skip around, but I think I'll get to the most interesting things. Because I'm talking about something that you may have never heard of before, I want to give a lot of background. So I'm gonna give you a lot more history than I am my own findings. So just to orient you a bit, here we're looking at the eyeball. I mean, it's funny, like there are so many talks today and many more tomorrow that are about the science of something like, you know, the qualia visual experience, like really amazing stuff. And I'm going to drill down to this one cell and what it does. I feel a little bit like I don't know, like a brick maker at an architecture conference, or I don't know, their architecture conferences, something like an architectural tour. But so just to orient you, here we see the eyeball. Here's an OCT scan. Omar showed you some of these earlier too. And back here at the back of the eyeball is the macula, and in the center of the macula is the fovea. And so here we're looking at a cross-sectional image of the fovea. If we zoom in on this yellow box here, we see the laminar structure of the retina in more detail. It's quite a beautiful thing, I think, the clinical OCT image. Next to it and aligned with it roughly, I have a diagram that I made of the various retinal neural layers, which, you know, transduce a neural signal from light and filter it in various ways as it's on its way to the brain. I say that this is approximately aligned because there's, believe it or not, some disagreement as to the attributions of some of these OCT structures that continue. We're not sure exactly where they come from, but we have guesses, and I'm going to be going with my guesses for the rest of this talk. Here on the right is a diagram of a rod photoreceptor and a cone photoreceptor, which will allow us to drill down even further to this cone outer segment, which is really going to be the topic of today's talk. As Omar mentioned, the cone outer segment and the rod outer segment is the site of phototransduction. So here we see on the left that diagram again, and just to the right of it, an electron micrograph of the discs of the outer segment. The outer segment is, as most of you know, like a stack of membranous discs associated you know, enveloped in a membrane, much like coins in a coin sleeve, like a bunch of sloppy coins in a sloppy coin sleeve. And on each of these discs, we see here, I think this is exactly the same cartoon that Omar showed. This is the phototransduction process. There are two important processes that are related to the stuff I'm going to talk about. One is phototransduction, probably the most important one. The other is the visual cycle. Both of these involve a number of important enzymes, which catalyze the transduction of visual signal and also provide homeostatic support for the photoreceptor or chorioretinal complex. And these enzymes fail or are mutated in a number of inherited retinal diseases and presumably implicated in other diseases that we don't understand as well, such as AMD. But these are the kind of the things that we're trying to study with optoretinography. Yeah, so the class of diseases that I'm most interested in my lab are age-related macular degeneration and then inherited retinal degenerations, especially those that affect photoreceptor function. And so because, it's like a case of if all you have is a hammer, everything looks like a nail, all I have is a way to look at outer segment behavior. So everything looks like photoreceptor disease. That doesn't quite make sense, but I want to just tell you what the clinical guidelines are for management or staging of these diseases now. And they're codified in various places. And I kind of chose some somewhat arbitrarily selected ones, but here from, this is from, what is it even from? I can't remember now. It's the official, it's from Retina. I think the journal Retina, they have these documents called PPPs, which are something, Omar, do you know? Preferred practice, yeah, plans, yeah. And there they outline how you're supposed to diagnose and stage these diseases. And for AMD, the structural approaches are fundus imaging, OCT, OCTA, fluorescein angiography. They mention adaptive optics in passing. It's not formally part of the guideline. And then on the functional side, they suggest, there's the standards like visual acuity, and then the Amsler grid, which is a kind of subjective way or psychophysical way of testing distortions in the image, in the retinal image. Inherited retinal diseases have a somewhat richer set of tools that are recommended. Similar kinds of structural tools, and on the functional side, they recommend microperimetry visual fields and ERG as well. To help understand what the cause of the disease is, or what the, in some cases, what the genotype is. I think this is probably a rapidly shifting field given how widespread sequencing is now. It's hard to say that, it's hard to imagine that inherited retinal degenerations will continue to be diagnosed with these tools. They're more likely to be diagnosed with sequencing. But in terms of staging them or assessing disease progression, or maybe most importantly, turning around clinical trials, we need other kinds of tools. The diagnostic's not sufficient. You need to know how bad is the disease and is it getting better. So there are my claim is that there's room for another type of tool, and in particular the kind of tool that I'm interested in developing is one that gives us some functional information from the retina, right? So you shine light in the retina, the image changes somehow, and we can take some figures of merit, quantitative figures of merit out of that response. We are not looking at function exactly. We're not looking at potentials. I mean, potentials is really, if you're interested in the function of the photoreceptor, it's right there in the membrane potential. We're not looking at potentials. We're looking at other things that the photoreceptor is doing, which we presume to be related to its function. And so there's a lot of work to be done there in understanding what it is we're seeing. As I go a bit through the history here, I just want to list some things that I'm omitting. I'm omitting changes in the amplitude of the image, that is how much light is coming back from the retina, omitting densitometry, changes in infrared scattering, and some paradoxical outer segment length measurements. But I wanted to put these citations up here anyway. They've been important for me in understanding what it is we're seeing. I want to go way, way back to get started. I put origins of optoretinography here, but this is not really the origins of optoretinography. These are just some images of squids that were caught out here off the coast of California in the 1940s. This is from a Fish and Game bulletin published by the state of California in, I think this was published in 1960 something, but these images are from the 40s. And, but this squid turned out to be a really important model of early neuroscience. You're probably familiar with it, most familiar with it in the work done by electrophysiologists, Hodgkins and Huxley, who figured out how action potentials work and whatnot using the same model. Down the hall from them were lesser known experiments done by Hill and Keynes around the same time, where they were looking at changes in the optical properties of the axon in response to stimulus. So they showed that this axon swells and shrinks when it propagates action potentials, which is like a seminal finding, and really much like what I'm going to show you now, although this took place 70 years ago. The first to show that the outer segment of the photoreceptor changes in length in response to visible stimuli were, well, Joe Corliss at Duke University kind of did. The most convincing evidence comes from this paper, Shabra and Cavagioni, in Nature 1973, published incidentally 50 years ago, this within a few days of today. So I think 50 years ago, 3 days ago is when that paper was published. On the right here, you see this modified kind of X-ray crystallography device. They lay a piece of frog retina over the top of this thing and immersed it in this solution, some kind of physiological solution. And then they shined an X-ray beam through it. And owing to the kind of quasi-crystalline structure of the outer segment, they were able to detect in the X-ray diffraction pattern, changes in the spacing of the discs, which they associated with changes in the length of the outer segment. So this is a very clever approach to the problem. We obviously can't do this in people. So the question is, what can we do in people? But the motivating hypothesis here is that the outer segment of the photoreceptor, rod and cone photoreceptor, changes in length in response to visible stimuli, and that these changes in length are very small, smaller than the wavelength of the light source, so extremely difficult to detect. So how do we how do we do it? We started off by using adaptive optics and OCT together. So you're all familiar, I mean, those of you who are in the Bay Area, this is one of the world's kind of epicenters of AO research, thanks to Austin Rorda's presence here. Adaptive optics, shown in this diagram on the left, measures and corrects the aberrations induced by the eye's liquid optics. OCT is a technique by which, I mean, you're familiar with OCT images, but it's basically an interferometer. It measures the time of flight of photons coming back from different layers in the retina and uses those times of flight as well as the associated amplitudes to build a cross-sectional image of the retina. I don't have a lot of math in this talk, except for this one slide, which shows a kind of critical thing about OCT. And you can just ignore this if you're not interested, I wouldn't blame you. But the spectrometer in spectral domain OCT measures a cosinusoidal fringe that's a function of wavelength and optical path length to the sample. The Fourier transform of this cosinusoidal signal gives you the amplitude and also the phase of the light coming back from that scatterer. Okay, so in OCT, we can see the phase of the light coming back from the retina. And now, unlike the optical resolution or axial resolution of the system, the phase gives us exquisite resolution in terms of length changes or movements of objects in the retina. There was some hint of this. This wasn't the question asked by these two investigators, Dita Bijeva in Wolfgang Drexler's lab and Vivek Srinivasan in Jim Fujimoto's lab showed they were looking at amplitude changes, which I said I was going to omit, but they had a couple of images in their papers on rat and rabbit retina, respectively, showing a suggestion that these layers might be rearranging in response to visible stimuli. So that was another hint that there was something to be seen here. And probably the most important paper came from Duke, from Joe Izat's lab, from a young scientist named Mike Choma, who showed that the phase of the OCT signal could be used to detect these tiny changes. He demonstrated it in a cardiomyocyte of chicken, showing the heartbeat or the beating of these very young cells in the fetal or the embryonic chicken. Yeah, and what this is one more equation I'll just mention. It tells you what the phase sensitivity of an OCT signal is. It is the square root of 1 over the SNR of the OCT, which can easily be 10,000, multiplied essentially by the wavelength. So we were talking about resolutions that are on the order of hundredths of a wavelength of the light source. Okay, now like a very quick history. In 2007, when I was a PhD student with Don Miller, the OCT systems were not fast enough to measure the kinds of changes we wanted to see. So we figured out a way to use the outer segment as an interferometer itself. The outer segment has these 2 reflectors, and if you interfere the light from those 2 reflectors with one another, you can detect changes in the length of the outer segment that are very, very small. So that was the first step. And a few years later, we showed that with an OCT system, an AO-OCT system, we can measure and stabilize the phase of the OCT signal enough to measure to in order to detect these small changes. The OCT system was still not fast enough, and it wasn't until Gary Huttman in 2016 built an OCT that seemed like kind of science fiction, an OCT system hundreds of times faster than anyone else's, that the first changes were observed. So that was in 2016. So here we're looking at changes in outer segment length, here averaged over these relatively large fields, as a function of time in response to visible stimuli. So we're seeing the outer segments lengthen or elongate, contract and elongate in response to visible stimuli. Since then, there's been a ton of progress, and I'm going to tell you about our contributions, a couple of papers here, just really these two papers, I think, all the time for. There are really 4 techniques that are in use to do optoretinography today, and I've got all 4 of them in my lab, but I'm only going to tell you really, again, like about the first 2. Maybe the third, if a moment permits. So here are some examples of AOCT images acquired with our AOCT system. These are two different, one is from the OCT channel, one is from the SLO channel. And you can see these now familiar mosaics of bright blobs that are the cone photoreceptors. These are, of course, 3-dimensional images. So you can look at them en face, as shown on the left and center, or you can look at them cross-sectionally. And if you look at them cross-sectionally, you see each cone is a pair of these blobby reflectors. And it's the phase of light coming back from those blobby reflectors that that we look at and pay attention to. Here we can see rods and cones in the system. And I'm going to show you in a moment what the respective responses of those 2 classes of cell look like. Here we have, I've just blown up that image, that en face image, to indicate a few of the cells. Here in the top left or upper sort of left, we see responses from individual cells. Down here, we see responses from individual cells as well. And what we see here is these 2 cone responses in blue and purple toward the bottom, and 3 rod responses in green, red, and black. So these are responses from single cells in the living human eye. And I think that they're on the order of 100 or even less, 50 nanometers in length. So one thing that's kind of tantalizing about these results is the respective properties of the responses of cones and rods. The cones look like they're less sensitive, but fast. The rods look like they're more sensitive, but slow, consistent with what we know about the sensitivity and bandwidths of the 2 systems. Both classes of cells show these very reproducible dose dependencies, which is consistent with this being a functional response. I mean, it's not proof that this is a functional response, but the fact that the response scales with dose over at least some range is encouraging and makes us think that we're looking at function. We built a faster AOCT in order to detect another aspect. I'll skip ahead here a little bit. Another aspect of the response, which is this initial contraction. So the cells elongate in response to light mainly, but over the first 10 milliseconds or so, they contract in response to light. This has been investigated further by Ram Sabason, who's attributed it to something like the early receptor potential. Now, AOCT systems, I will say a little bit about the third approach. AOCT systems are big and cumbersome. They require multiple people to operate. They're expensive. They're temperamental. They're always failing. They don't work on a lot of subjects. And so we wanted to build something that was more clinically viable, perhaps. And so we decided to build a conventional OCT system out of components that are very much like those you'd find in any clinical OCT system, and see whether we could measure similar changes. So this is what we've been working on for the last year or so. There's a picture of the system in the center. It's meant to look very simple. I hope it does. And on the right is an example B scan from the system. The key difference here is that, with the AO systems, what we do is segment individual cells, and through motion tracking, we track them over time. And we're always looking at these 2 reflections from the same cell. With this method, we are doing B scans only, so it's not volumetric imaging, and the retina may be moving. So there's no guarantee that we're looking at the same cells over time. So rather than looking at the positions of the scatterers, which can vary from cell to cell and give you spurious signal, we're looking at the velocities of the scatterers, which we assume to be correlated highly among the stimulated region. So that's the key difference, and we're getting very nice results here. These are 3 different subjects at 4 different retinal eccentricities. We seem to have these kind of reproducible patterns of contraction and elongation that are consistent with the AOCT measurements. And I think that's where I want to stop. Yeah, that's where I want to stop. I'll go to my thank you page. Thank you.
Yeah, question?
Thanks, Omar Maru. Thanks, that was fantastic. And you talked about using it in inherited retinal diseases, and I saw the slides at the end. Any early results on that?
Yeah, thanks for asking, Omar. Now I have an excuse to go back to the slide. Yeah, we have there. I mean, I really want to show them because they're so early and they're so noisy. It's very hard to say much of anything yet. We have some preliminary looks at ABCA4, Stargardt's disease that Omar mentioned, as well as an RP variant whose gene mutation I can't remember off the top of my head. And AMD, and we see some changes in the response of the photoreceptors. I mean, there are some intriguing details here, like the ABCA4 patient. It's very noisy, but we have a number of scans from that patient, and in all of them, we see the contractile portion, but not the elongation, which suggests that the mechanism underlying the ERP, which is really this dipole shift in the outer segment, it's kind of esoteric. The early receptor potential might be there, like the photopigment is present, but there's just something missing, something else that's missing in order to lead to the elongation. Yeah, I don't know. It's tantalizing, but very preliminary.
Thanks.
One more question? Thank you very much.
Yeah.